Hi, I'm Mike DeLong, and today I have the privilege of discussing our new JACS article entitled Evaluation of Data Requirements for Animal-Derived Wound Care Devices, Limitations of the 510K Pathway. This topic became relevant after the recent FDA panel meeting to assign classification for these devices in October 22. The panel unanimously recommended a Class 2 designation, which relies on the 510K clearance pathway, where manufacturers must demonstrate that their product is substantially equivalent or similar to another device already on the market. However, despite this recommendation, the panel did voice concerns about potential clinical data limitations of using this pathway. We decided to investigate these concerns by evaluating the publicly available clearance statements on the FDA website, as well as the PubMed index literature for all animal-derived wound care devices. Of the 132 devices cleared through this category, 114 had publicly available information. Most of the products used animal components from bovine or porcine sources, although many different animals were represented. Preclinical biocompatibility testing were required in 75% of products and were leveraged from a different submission in another 9% of products. Preclinical animal wound healing testing was only required in 15% of products and was leveraged in another 4% of products. We found that none of the products were required to perform clinical wound healing testing in human patients, which is not unexpected for a device cleared through the 510K pathway. In the PubMed index literature, we found that 73% of products had no clinical studies to support their performance. But on the other end of the spectrum, a few products, about 5%, had over 10 supportive publications. We conclude that wound care is a heterogeneous and varied clinical practice with a multitude of potentially preferable device attributes, but that minimal clinical data are available through the FDA clearance process to inform device selection. Therefore, any claims about effectiveness for wound healing or other properties are likely to require independent clinical validation, which is often absent in the primary literature. Therefore, practitioners are left to their own expertise and experience when selecting a device.